Important questions from blood physiology. What is the total blood volume in a body? 5 to 6 liter, that is 8% of the body weight, or 80 ml per kg body weight. Enumerate functions of blood. Respiratory, transport of gases, oxygen from lungs to tissues and carbon dioxide from tissues to lungs. Nutritive, conveys absorbed nutrients from the GIT to the tissues. Excretory function, transport metabolic waste to the kidney, skin and intestine for the removal. Homeostasis, maintains the homeostasis of electrolytes. Regulation of body temperature, as water in blood has high specific heat, high conductivity and high latent heat of vaporization. Chemicals for communication, protects against blood loss through hemostatic clotting mechanism, protects against diseases through WBCs and antibodies. How serum differs from plasma. Blood after clotting shrinks and gives out serum. It is thus plasma minus fibrinogens and clotting factor 2, 5 and 8. It has higher serotonin, that is 5-HT, content due to the breakdown of platelets. Plasma is clear, straw colored portion of the blood. If anticoagulated sample of blood is allowed to stand in a tube, on settling it separates out into cells and plasma. What is origin of plasma protein? In embryo, it is mesenchymal cell. In adult, albumin, fibrinogen and prothrombin from liver, globulin and tissue macrophages systems, plasma cells, globulin from tissue macrophages system, plasma cells or lymphocytes. Names forms of cell proteins based on Whipple's experiment. Fixed cell protein or dispensable cell protein not mobilized even when serum plasma protein level is less than 2 grams per deciliter. Dispensable reserve proteins called upon energy purposes in starvation and malnutrition, etc. Example, endogenous protein from skeletal muscles and glands. Liable reserve protein, they move out immediately into the bloodstream and compensate for protein loss such as hemorrhage, burns, etc. Main sites are liver and reticuloendothelial system. List functions of plasma protein. Helps in coagulation of blood. Helps to maintain colloidal osmotic pressure across the capillary wall. Normal colloidal osmotic pressure exerted by the proteins is 25 to 30 milligrams of mercury. Helps in maintaining viscosity of the blood at a low level since 80% of the total plasma protein concentration is due to albumin. Provides stability to the blood due to the presence of globulin and fibrinogen. Helps in maintaining the acid-base balance in the body because plasma protein are amphoteric in nature, that is they behave as acids as well as bases depending upon the condition. Their buffering capacity is 1/6 of the total buffering capacity of the blood. Transports and reservoirs of hormones, drugs and metals. Provides immunity due to the gamma globulin fraction of the plasma protein. What is colloidal osmotic pressure and how it is produced? Colloidal osmotic pressure is exerted both by the crystalloids, urea, sodium and glucose, and colloids like plasma protein. Colloidal osmotic pressure is due to the plasma proteins alone is known as oncotic pressure. 80% of the colloidal osmotic pressure due to the albumin because of least molecular weight and maximum concentration. Determine the rate of exchange of fluid along the capillary. What are Starling forces? That I will be saying later. How does hypoproteinemia produces edema? Hypoproteinemia decreases colloidal osmotic pressure, increases filtration at the arterial end, decreases the absorption of the fluid at the venous end. Abnormal collection of the fluid in the interstitial space is known as edema. What will happen if iron in heme is present in ferric form? The condition is known as methemoglobinemia. Disadvantages: it cannot unite reversibly with gas or gaseous oxygen. It is dark colored, and when present in more than 1.5 grams per deciliter in circulation, it resembles cyanosis, blue discoloration of the skin. Thank you.